All right, so these notes are for section 4, chapter 5. The objectives are to compare and contrast binary fission and mitosis. And so we're going to talk about prokaryotes and how they divide, which is different than how your cells divide. So I have binary fission. That's going to be a prokaryote thing. And I'm going to have mitosis. We already talked about mitosis. That's what happens in your cells, in a eukaryotic cells. And again, we're going to compare and contrast once we learn about binary fission. Essentially, prokaryotes are a lot simpler than you. Prokaryotic cells are simpler than eukaryotic cells. So it's a simple process. It's a lot easier to do, right? At the end of the day, a prokaryote doesn't have any organelles. A prokaryote only has one chromosome to divide. So that eukaryotic cell has a lot more things to do just because it has more chromosomes. Your cells have 46 chromosomes. That eukaryotic cell has a nucleus that needs to be dissolved and recreated. So we're going to compare and contrast those things, but we're also going to talk about how some eukaryotes can reproduce through mitosis. Otherwise, asexual and sexual reproduction pop up as a topic. Technically, sexual reproduction is not in this section. I just put this definition in here for comparison so that you can kind of look at one versus the other. But fill in blanks, that sexual reproduction involves the joining of gametes from two parents. So you have gametes, you got two parents, and you end up with genetically unique offspring. Right? We should be familiar with that because humans reproduce sexually. And you know that you got genes from each of your parents, but although you might look similar to one or both of your parents, you don't look exactly the same as either of them because you inherited a mix of traits, right, that made you unique. You're not the same as any of your siblings either, right? Not exactly the same, not unless you happen to be identical to a twin, but that would be a whole different situation. Otherwise, asexual reproduction, you have genetically identical offspring, and there's only one parent, right? That's what we're going to be talking about when we talk about binary fission, because technically when a bacteria divides, it is a reproduction event. Those are two separate cells that are their own organism. So binary fission, we need to fill in a blank or two here for our definition. But really, I'm going to draw a picture for this process to try and represent it. It's just a little bit easier for me to do it that way. So you're going to need to draw this in your notes, probably at the bottom or something like that, wherever you can fit it. But I also, in class, had your friends, your classmate, your peers, come up with some questions. Remember, that's what we're using the left margin for. So make sure you're thinking about questions you might ask from these notes, because I might pick on you guys in class when you come back, right? So you can ask those questions and make sure you get them answered. Otherwise, to do this process, binary fission, right, I'm going to start with a bacteria cell up here, right? So doesn't have to be perfect or anything, just want to try and make sure that connects. But this little circle that I'm going to draw inside the bacteria is actually going to be its DNA. Remember, that could be an organelle if this was a eukaryotic cell, but there are no organelles in prokaryotic cells. So that's a piece of DNA. Prokaryotes actually have circular DNA, which is different than your linear DNA. Right? Plus, they only have one piece of DNA, just that pink DNA that I drew in there. So the first thing that would need to happen before a cell could divide, a bacteria cell could divide, is that one piece of DNA needs to be copied. So I have a piece of pink and orange in this cell now, and both of those pieces of DNA are going to be attached to the membrane, right, directly to that membrane of the cell. And basically, that is the hardest part of this whole process, copying the DNA and attaching it to the membrane. Because after you've done that, there's really not much left to do. The cell just needs to grow so that it's big enough that it can divide. And as it grows, automatically the DNA is going to be separated. One piece is going to go to one side of the cell. The other piece will just move its way to the other side of the cell. And at this point, the only thing that needs to be done to finish the process is I'm going to need to have some furrowing. I'm going to need to do some kind of cytokinesis where the cell is going to divide into two cells. Right? So I start pulling on the drawstrings for that hoodie, and I pinch that cell in half so that I have one cell over here, another cell on this side. They both have identical copies of DNA, right? That pink versus the orange. I only made them different colors so that we could see that they're right, different from each other, although there are identical copies of one another. Right? So each cell is identical to the parent. Otherwise, all things are in the words here as well, right? But it's really simple. It's not very complicated. Once the DNA is replicated, that's step one, you got to attach it to the membrane. The cell's going to grow and elongate. At that point, all you need to do is pinch off that membrane, and you're going to have two new cells. The only thing that I didn't talk about yet is how you're going to have to build new cell wall to finish off the process. So I can add that to my picture as well. Right? There's actually a cell wall around this original cell. And there's going to be a cell wall around this cell as well. It's kind of hard to draw using this tablet. But otherwise, I need to complete that cell wall because I pinched the membrane off. But that doesn't mean that I built new cell wall to completely surround these two new cells. So that's the last step of the process. I need to rebuild and make new cell wall to finish off this process, right? So I'm starting to build, use maybe a dotted line, new cell wall to finish that off. So that was the last step, the only thing I had left out, but otherwise visually you could see how that would work. So binary fission and mitosis, you have to compare and contrast those things. So I'm going to organize some information in a table here, kind of. This is the way I kind of like to do it. So things that they have in common are in the middle. Otherwise, this is binary fission on the left, mitosis on the right. Just abbreviating that. Binary fission happens in prokaryotes. Mitosis is in a eukaryote. Both take one cell and turn it into two cells. And both create identical daughter cells, right? We did talk about how binary fission could be used for reproduction and is a reproduction event for a bacteria cell, whereas mitosis is typically just going to make new cells in a multicellular organism. So you make new skin cells to replace old ones. All right, so otherwise, what else we need in here? I can say that binary fission is simple and mitosis is complicated. And again, I already gave you reasons for that. Essentially, binary fission, pretty simple process because you don't have a nucleus to deal with. There are no organelles in a prokaryote, right? You don't have to dissolve or recreate a nucleus, which is the whole, like that's half of what you're supposed to be doing in prophase and telophase in mitosis, right? Otherwise, I also talked about how there's only one chromosome, one piece of DNA in a bacteria cell, whereas there's many chromosomes in a eukaryote that you need to divide, right? For us, for example, for humans, there are 46 chromosomes, right? So that number could change, but just since this would be relevant for you in your skin cells when they're mitotically dividing, right, that new cell needs 46 chromosomes just like the original cell. And you can't just attach those randomly to the membrane and hope that the right 46 go to one side and the other 46 copies go to the other side. You're going to end up with some cells that don't have the right mix of DNA, and that would be bad. So there's a reason why it's organized, why you need the prophase, metaphase, anaphase, telophase to separate those chromosomes appropriately. But otherwise, if you have all this information in this table, you can add more things too. But these were things that we came up with in class, so I'm guessing this is pretty comprehensive. We also talked about the advantages of asexual reproduction. So firstly, they can multiply exponentially, right? So again, for a visual purpose, right, one cell divides to make two cells. But then those two cells divide to make two more apiece, which makes for 4 total cells after 2 rounds of division. Now, the one thing I'd point out, just because sometimes that's lost on people, right? Remember, once this original cell divides, these 2 new cells exist, and it doesn't really exist anymore, right? So 1 turns into 2. Those 2, once they divide, don't exist anymore, but I have 4 new ones to replace them. So my total as I go from 1 to 2 to 4, and you can continue with that pattern. 4 turns into 8, 8 turns into 16, 16 turns into 32, right? It just doubles, which is why instead of actually having to draw out all the cells that are going to be formed, it's much quicker to just use a little bit of math. So I can use this equation up here. I can use this 2 to the x power, x being the number of rounds of division that you've had. So if I wanted to know how many cells there'd be after 5 rounds of division, 2 to the 5th power will give me that answer. So again, you just work that out. Again, 2 to the 5th power just means 2 times 2 times 2 times 2 times 2, right? I have 5 2's. If I multiply that out, I just break it down a little bit. There's a couple 4's. 4 times 4 is 16. 16 times 2 is 32. After 5 rounds of division, there are 32 cells, right? Now, I can show that visually again, and it does work for all situations. 2 to the 0 power, if I haven't divided, anything to the 0 power is just 1, right? 2 to the 1st power is 2. 2 to the 2nd power is 4, and I'm still going to get that pattern. 1, 2, 4, 8, 16, 32 was for round number 5. After 10 rounds of division, right, there are actually going to be 10,000, and I believe it's 24 cells. You might have to check that out on your calculator. It's 32 times 32, basically, to get that answer. After 20 rounds of division, there are actually more than 1 million bacteria cells. So in class, I talked a lot about how bacteria can multiply quickly. Right? It doesn't matter, right? You should still do it, but it doesn't matter how many times you brush your teeth. If you can't kill every single bacteria in your mouth and you can't guarantee that new bacteria don't get in there, right? They're going to multiply quickly overnight, and there will be millions in there for you to deal with again, right? There's a reason why you wake up with dragon breath some days, right? So bacteria can multiply very, very quickly. They can go from 1 to 1 million overnight, right? So that's always going to be an advantage for them. No matter what the organism, if it's reproducing sexually, right, it could never keep up. So even though a fish can have 2,000 eggs, right, that's still not going to happen every day, right? They're not going to be able to do that on a consistent basis and make 1 million new eggs every day. So no sexually reproducing organism could ever keep up. Bacteria divide just so quickly that it's definitely an advantage for them. The second advantage is that all cells can have offspring. You don't have to find a mate, right? So those bacteria cells don't worry about whether there's another bacteria cell around so that they can have offspring because they don't need it. Whereas a female fish can lay thousands of eggs, but if there's no male to fertilize those eggs, then there's not going to be any offspring anyway. So again, those are our advantages for asexual reproduction. Whereas technically there are disadvantages as well, right? If you have genetically identical offspring, right, they're not going to be able to can't respond to that changing environment very well, which is why when you rinse and gargle with Listerine for a minute, you can kill a lot of bacteria in your mouth, right? You change those conditions that those bacteria live in, and they can't cope with that change, they're all going to die. Because if one dies, they all die. They're all identical, so if it's bad for one, it's bad for all of them. Whereas sexually reproducing populations actually have an advantage there, right? We can survive, and sometimes we're even going to thrive in those changing environments. So that's why we want those different traits. We want to all be different so that we can adapt, so that we can evolve in those situations, right? I also mentioned in class that, it seems like being identical is bad for bacteria, but it's not really that bad for them, right? If you haven't noticed, again, you can use Listerine every day, but you're still going to end up with bacteria in your mouth, right? Unless you can guarantee that you kill every bacteria, even the ones hiding between your gums and between your teeth, right? There's definitely going to be one left that's going to rebound to millions overnight. So really, for them, it's not that big of a deal, right? It does suck that a lot of organisms die off, but as long as you have one survivor, you can rebound, that population can rebound very quickly, right? So it's a disadvantage, but bacteria have been around for millions of years, right? One of the sayings I like to use to refer to this, right? If it ain't broke, don't fix it, right? Bacteria are doing fine. They've survived for a long, long time. And they actually can evolve. They just have to depend on mutations to get new traits. So again, as long as you don't kill all of them, and as long as every once in a while they have a mutation that helps them get a new trait, bacteria do just fine. So we do have the advantage. Sexually reproducing populations have the advantage in terms of evolution, but that doesn't mean that bacteria are going to go extinct any day soon. So in this last slide, really our main focus is just to make sure that we know that some eukaryotes can reproduce through mitosis, right? So this is not binary fission. This is not a bacteria cell, right? Some simple eukaryotic cells can use these processes, so you need to be able to recognize these in a homework quiz and or quiz test. But budding, fragmentation, vegetative reproduction, those are all example of mitotic reproduction, right? So that yeast we use in the experiment actually they reproduce using a budding technique, right? Now, just to give you an idea of what that's like, it's not really possible, but if I was to grow a Mr. Morris off of my back and then it jumped off and was running around and that's my kid, right? That would be an example of budding, right? Obviously not realistic. There aren't any Mr. Morrises running around like that, but that's the idea. That's what would happen. Fragmentation, right? I was informed earlier that there's an episode of SpongeBob even that Patrick's arm got cut off and he regrew his arm because that's something that sea stars can do. But the arm regrew a new Patrick, right? And that's actually a real thing, right? Some organisms, if you have a large enough chunk that breaks off of them, that chunk can turn into a whole new organism. So it's most likely very simple organisms like flatworms, but sea stars can actually do that too. The vegetative reproduction, some of you may already be aware that if you take part of a plant, you can move it to a new location and grow a new plant. Right, so strawberries, potatoes are two examples of that. Otherwise, that last example is kind of a weird one, but it is what it is. technically there are some things that can actually reproduce both sexually and asexually. So sea anemone are like that. So you would know what that is because you've seen Finding Nemo. Nemo lived in an anemone, and so they're that, they're those, they live in the ocean. They have these tendril-like fingers, right, that are just catching stuff as it goes through the water to feed on, right? But they're also poisonous, not to clownfish, which is why clownfish can live in them, right? But sea anemone, right, can actually reproduce sexually or asexually. You can imagine that if you had an egg that was fertilized for one of these and it floated off in the ocean and decided to deposit itself on the ground where there were, on the bottom of the ocean where there were no other anemone around, it would have a tough time reproducing sexually. So it has to have some other option. But generally speaking, if there was another anemone around, it would reproduce sexually. So this organism has the ability to do both because obviously there are advantages of both. So those are all the things we needed to do in the notes. Take care of a homework quiz and we should be fine.